Hello students, today we talk on the theories to explain short distance translocation. In previous video, I discussed about the solute transport and structure of the sieve tubes and sieve plates. So mostly the translocation of the solutes is takes place through the phloem. So this phloem is contains the different parts like sieve plates, sieve tubes, then companion cells and parenchyma cells. And according to the mechanism of translocation or phloem conduction, there are two theories. One is to explaining the short distance translocation, and another one is the long distance translocation. So now start the short distance translocation. So this for explaining this short distance translocation, four different theories are mentioned. In that, first one is the protoplasmic streaming theory, second is the transcellular streaming theory, third is the contractile protein streaming theory, and fourth one is the electroosmosis. So we're looking one by one. First is the protoplasmic streaming theory. So it explaining for the short distance translocation. According to him, the protoplasmic streaming occurs on the sieve tubes elements of phloem solutes along with the protoplasm, that is cytoplasm, are carried from one end to the other end of the sieve tubes. This theory is proposed by the scientist De Vries. At other end of the sieve tube, solute diffuses to next tubes. This was supported because it accumulated for simultaneous movement of solutes in both upward and downward direction in same sieves tubes, and the factors like temperature, oxygen deficiency, which affect the protoplasmic, also affect the transport of the solutes. So this is the first protoplasmic streaming theory. In that, the scientist De Vries mentioned that the these mostly this protoplasmic streaming theory, it is occurring in the sieve tubes elements of the phloem, and the solutes along with the protoplasm are carried from one end to the other end of the sieve tubes, and at the other sides or other end of the sieve tube, solute diffuses to the next tube, and this was the shows the movement of solutes in upward as well as downward direction in the same sieve tubes. And also they mention about the effects of temperature and oxygen deficiency that will be also affected on the transfer of the solutes. So the objective of this theory is that is the strongly objection against this theory is that the protoplasmic streaming has not been observed in mature sieve tubes elements. So regarding this theory, one objection, that is these protoplasmic streaming that is not observed in the mature sieve tube. जो mature sieve tubes है उसमें हमें प्रोटोप्लाज्मिक स्ट्रीमिंग दिखाई नहीं देता है. Then about the second theory, that is the intracellular streaming. This theory was proposed by scientists Canny and Thien. This theory is also callous this theory is also called reconstruction of the protoplasmic theory. That means the cytoplasmic strands traveling sieve tubes elements in petiolar tissue. The movement of solutes from one sieve tube to other, the solute moving in opposite direction in adjacent strands in same sieve tubes elements. सो जो प्रोटोप्लाज्मिक स्टैंड्स में हमने देखा है कि अपवर्ड और डाउनवर्ड डायरेक्शन में जो भी सोल्यूट्स है उसका मूवमेंट होता है. सो दिस इज द सेम है, बट some points or some views are added in this theory. That's why this theory is called as the reconstruction of the protoplasmic theory. In that they can add it about the जो भी intercellular spaces है, that is the जो cytoplasmic strands रहते हैं, these cytoplasmic strands are also translocate the solutes. That's why the movement of solutes are also takes place from the one sieve tubes to the other, and that will be helps to the sieve tubes. प्रोटोप्लाज्मिक थ्योरी में हमने क्या बताया कि वहाँ पे जो भी sieve tubes है, sieve tubes वहाँ का जो solutes रहता है, solutes को ट्रांसपोर्ट करती है. साथ में इंटरसेलुलर थ्योरी में क्या बताया है कि जो भी साइटोप्लाज्मिक स्ट्रैंड्स है, दिस साइटोप्लाज्मिक स्ट्रैंड्स आर आल्सो ट्रैवल्स और आल्सो ट्रांसलोकेट द सोल्यूट्स with the sieve tubes. Then in the third theory, that is the contractile protein theory, this explain short distance translocation, and it proposes that is the network of interlinked microfibrils in sieve tubes. Such a network accelerates in some manner of that moving flagella in organisms. That is, particles attached to this microfibril and moves with the bouncing motion. तो अभी ये थर्ड थ्योरी में उसमें क्या बताया है कि जो sieve tubes में कुछ माइक्रोफाइब्रिल्स का नेटवर्क रहता है. माइक्रोफाइब्रिल्स इंटरलिंक्ड कंडीशन में हमें उसमें दिखाई देते हैं. जो दिस माइक्रोफाइब्रिल्स और हिज माइक्रोफाइब्रिल्स नेटवर्क्स will be helpful in the moving of the flagellas in organisms, or also they can help in the movements of particles in very बाउंसिंग मोशन में. ये जो भी पार्टिकल्स है, पार्टिकल्स को मूव करते हैं, और जो भी सॉलिड पार्टिकल्स है वो उसको ट्रांसलोकेट करते हैं थ्रू द microfibrils. Then fourth theory is regarding the electroosmosis. So this explain short distance translocation. According to this theory, the current of potassium ions passes through sieve pores by electroosmosis. The sugar molecules adhered tightly to the ions are coming along with them. Then compute current of K plus ions has circulatory pathway on ATP derived from neighboring cells is utilized to derive potassium ions. Thus each sieve plate is an electroosmotic pumping station which boosts mass flow of sugar solution at intervals along the sieve tubes. So this is also regarding the electroosmosis, or in that these potassium ions plays the role of the, or it will be the pumping station, and they can be provides the sugar solution after the intervals along with the sieve tubes. Sugar solution, that means they provided the ultimately they provided the energy in the form of ATPs. So this will be helpful in the uh, translocation of the solutes. In that there is the potassium ions which are present in the sieve pores of the sieve plates, and they can uh, uh, there some sugar ions which are, or sugar molecules which are tightly bind with these pores, and uh, if they have in the potassium ion has the adders with the sugar molecules with the potassium ions, and they can provide the ATPs, and ultimately the ATPs will be utilized with this potassium ion, and this potassium ion plays as a pumping station, and ultimately they provided the energy through these sugar molecules or sugar solutions after some intervals. But the limitation of this theory, that is the lack experimental evidences. Um, mm, on this electrosmosis, there will be not much work has been done, so the experimental proof is not available on this concept. That's why this is one of the limitation of this theory. So for short distance translocation, uh, here four theories are mentioned. In that, first one is the protoplasmic steaming theory. प्रोटोप्लाज्मिक स्टीमिंग थ्योरी में शॉर्टली आप क्या याद रखोगे? दिस थ्योरी इज प्रपोज्ड बाय द साइंटिस्ट डेवेरिस ने प्रपोज किया है. दे शुड मेंशन अबाउट दिस protoplasmic or sieve tubes elements of phloem with the protoplasm. They can carry solutes from one end to the other end of the sieve tubes in the both the direction, that means upward as well as the downward direction. But and the some factors like temperature, oxygen deficiency, these are also affected the translocation of the solutes. One of the objection of this theory, that is this theory or protoplasmic steaming are not observed in the mature sieve tubes. Then in the second intracellular steaming theory, that is the also proposed by scientist Carey and Thien, and they can reconstruct it of the protoplasmic theory, and they can mention that the cytoplasmic strands are also involved in the translocation of petiole with the sieve tubes or with the protoplasms. Then and the third contractile protein theory, in that theory it's explaining about the microfibrils in sieve tubes. These microfibrils forms the network of the interlinked cells, and they can helpful in the moving of the flagellas in organisms, and they can uh, uh, the particles will be attached with this microfibril, and they can move with the bouncing manners. Then fourth theory is electroosmosis. So this electroosmosis, in that some in the sieve pores some potassium ions and the sugar molecules are tightly present, and these potassium ions will circulating the ATPs, and they can attach with the potassium ion So these sieve plates is a electroosmotic pumping station, and they can provide the sugar solutions uh, uh, at some intervals to the sieve tubes. But the limitation regarding these electroosmosis, that is the no evidence of experiments. That's why this is one of the evidence of this theory. So for short distance translocation, these four theories are mentioned, and that will be proved about the translocation of solutes in short distance. So now I stop here. In next video we talk about the Munch hypothesis, that is regarding the long distance translocation of the solutes.